All right, well, welcome everyone to today's ASBMR member spotlight session. This is a series that was implemented by the ASBMR Diversity, Equity, and Inclusion Committee. And this series aims to highlight the research of ASBMR members in a collaborative setting, in particular to shine a light on research being done by ASBMR members belonging to groups historically underrepresented in biomedical research. Today, we have a special spotlight session in which we are getting to highlight the work of two of our 2021 underrepresented minority mentor awardees. This is a new program implemented this year. And so we are pleased to welcome Marquia Beau, a PhD candidate at the University of Florida, who will be presenting comparing subchondral bone remodeling following either anterior cruciate ligament or medial meniscus damage in the rat, as well as Ernesto Solorsano, PhD candidate at Northeast Ohio Medical University, will present detrimental effects of low aerobic capacity on bone metabolism. Each presentation will take approximately 15 minutes each, and following each presentation, there will be an opportunity for Q&A and discussion with the audience. As a reminder, we'd like to ask you to please keep yourself muted throughout the meeting during the presentation. However, you are welcome to unmute yourself and put yourself on camera during the questions or comments following each presentation. You may also submit your questions in writing via the chat feature. And so with that, I would like to invite Marquia to introduce herself and begin her presentation.
Thank you so much, Lauren. We've been emailing back and forth for quite some time, so it's really nice to see you and put a face to all of the communication. I will share my screen. Can everyone see my screen?
Yes, we can see your screen.
Okay, perfect. Thank you. So as Lauren mentioned, my name is Marquia Beau. I am a PhD candidate at the University of Florida in the Orthopedic Biomedical Engineering Laboratory. And today I will be presenting my work on comparing subchondral bone remodeling after anterior cruciate ligament or meniscus damage in rats. So to begin, the clinical presentation of osteoarthritis involves changes at the osteochondral junction. In a typical clinical radiograph, this may include the appearance of osteophytes, which are bony outgrowths at the joint margin, or subchondral bone sclerosis, which is thickening of the subchondral bone plate. Now, these changes, although represent subchondral bone remodeling, represent these changes at the end stage OA. And although there is a wide array of clinical data to support findings with bone changes at end-stage OA in clinical patients, there are few preclinical studies that examine bone changes at this similar stage. Even fewer preclinical studies compare bone changes between different preclinical models. Since bone changes have been linked to pain in OA patients, there is a need to better understand what happens in preclinical OA at end-stage OA. With this in mind, we had two primary questions. The first was, what are the characteristics of subchondral bone remodeling at the advanced stages of OA in rat meniscal and anterior cruciate ligament injury models? And two, are the bone changes we're seeing between these different rat models of OA unique? The study that I will speak about today compares subchondral bone remodeling in different rat OA models to examine the differences in the changes at end stage. To do this, we had 32 male Lewis rats split into 4 groups, depending on which procedure they received or which model of OA or control they received. In the first group, which is the medial collateral ligament transaction or the medial meniscus transaction group, or the MCLT plus MMT group, the joint of the rat was open so that the medial collateral ligament was visualized. Once it was visualized, we transected the medial collateral ligament, and the medial meniscus was also transected. In the skin incision group, the medial collateral ligament was visualized but was not transected. Following this, animals in both groups were imaged via ex vivo micro CT. The last 2 groups were the non knee injury group or the Nicky group. And in this model of OA, we had the affected limb of the animal placed in a custom fixture as shown here. And once this was done, we applied a compressive load to the limb and the ACL was ruptured. The control for this group was the last group of animals, the Nicky sham group, where we placed the limb of the animal in the custom fixture, but no load was applied to rupture the ACL. Following this, as before, the bones were imaged via ex vivo micro CT. Now we focused on bone changes in the epiphyseal region of tibia, specifically consisting of the subchondral bone plate and the subchondral trabecular bone, which together comprise the subchondral bone, or all bony components of the knee distal to the calcified cartilage. Here, pathological changes in subchondral bone are important to study because they can provide insight into the mechanical environment of the OA joint, or changes to the major load distributing tissue, which is a subchondral bone in the knee, may help us to further understand OA-related pain and disability. With this knowledge, we may be able to develop future treatment measures for OA. Now, to begin, we wanted to look at the qualitative bone changes to really visualize what was happening to the bone across the different rat models. We found that in both the MCLT plus MMT and the Nicky groups, there was thickened trabeculae. There was actually thinning of the subchondral bone plate in the Nicky group, which was an unexpected change, and thickening of the subchondral bone plate in the MCLT plus the MMT group, which is what we expected from different clinical studies that showed subchondral bone sclerosis at end stage. There was also the presence of bone marrow lesions in both groups. And we found that in the contralateral limbs of all of the animals in both groups, and in the control groups, none of these changes were present. Looking further at cross-sectional images of these changes, we saw the presence of pre-osteophyte formation, bone marrow lesions, and osteophyte formation. Looking further at the quantitative outputs, we saw certain trends in different trabecular measures and subchondral bone cortical measures. So to begin, we saw that the plate thickness actually increased in the injured limb of the MCLT plus the MMT group compared to the contralateral controls, and it significantly decreased in the Nicky group compared to the control group. Since an increased bone plate thickness indicates subchondral bone sclerosis, we concluded that decreases in the subchondral bone plate thickness in the Nicky model were not characteristic of the end-stage OA changes that we expected. Moving on to percent bone volume, we found that percent bone volume actually increased in the MCLT plus the MMT group compared to control groups, and significantly decreased in the Nicky group compared to controls. It also was significantly higher in the MCLT plus the MMT group compared to the Nicky group. So what do these shifts actually tell us? Since percent bone volume tells us how much bone is actually present in the region of interest, we concluded that the percent bone volume increases in the MCLT plus the MMT group verified increases in the average thickness of the residual trabeculae. With this in mind, increases in percent bone volume and trabecular thickness may indicate that the sclerosis that we were seeing in the subchondral bone plate developed throughout the trabecular bone. Percent bone volume also decreased in the Nicky group, and this verified decreases in the trabecular thickness. For trabecular number, we observed that trabecular number decreased in the injured limbs compared to the contralateral controls in the MCLT plus the MMT and Nicky groups. And the trabecular number was significantly higher in the MCLT plus MMT group compared to the Nicky group. Decreases in the Nicky group compared to the Nicky control was largest. Since decreases in trabecular number indicate bone loss, we concluded that in the MCLT plus the MMT and Nicky groups, they both experienced this pathological bone loss, and the Nicky group actually displayed more severe bone loss compared to the MCLT plus MMT group. For trabecular thickness, we observed that the thickness increased in the MCLT plus the MMT group compared to controls, and significantly decreased in the NICI group compared to the control group. There was also a wide variance in the medial compartment of the NICI group with trabecular thickness. And the largest increases in thickness we saw were between the MCLT plus the MMT animals. Since larger increases in trabecular thickness could indicate 1, possible pathologic or anabolic effect, and 2, smaller trabeculae was resorbed or larger trabeculae dominated the analysis, we concluded that in the MCLT plus the MMT group, there were more severe changes which could be associated with end-stage OA, or the anabolic response to shifts in the mechanical environment of the joint. There was also a wide variance in the medial compartment of the NICI group, as I indicated earlier, and this may describe some possible nondescript changes. For trabecular separation, we observed that separation significantly increased in the MCLT plus the MMT and the NICI group compared to the control group, and it was significantly higher in the NICI group compared to the MCLT plus the MMT group. Since trabecular separation or a higher trabecular separation indicates a greater distance between trabeculae, we observed that the increased separation in the MCLT plus the MMT and the NICI groups support our findings of reduced trabecular number, and this increased separation in the NICI model indicates signs of severe OA. For trabecular pattern factor, we observed that the NICI model showed the greatest difference between the injured and the contralateral limbs. And the pattern factor was significantly increased in the MCLT plus the MMT and NICI group compared to the control groups. It was also significantly higher in the NICI group compared to the MMT group. Since the increased trabecular pattern factor indicates a less connected trabecular structure, we concluded here that both the MCLT plus the MMT and NICI groups show changes consistent with end-stage OA, where changes were more pronounced in the NICI group. So both this qualitative and quantitative data motivate the need for analysis of bone at later stages of rat OA, where we concluded that the bone changes in the NICI group were in fact different from the bone changes in the MCLT plus the MMT group, and bone changes at the extended time point show more severe remodeling in the MCLT plus the MMT model, and some unexpected changes in the NICI model. Now, to understand why these changes are different requires a closer look at the events that precede bone changes. Here, we're talking specifically about the gait abnormalities that may drive bone changes. Since subchondral bone remodeling is an error-driven adaptation, our future work will examine the relationship between bone changes and gait abnormalities that precede and/or follow bone remodeling. With that, I'd like to thank my lab, our collaborators, and everyone who contributed to this work. And I can take any questions.
Thanks, Markia. I will invite people to submit your questions via the chat, or please come off of mute and you can turn on your video and ask your questions to Markia.
Very nice presentation, Markia. So I have a question. How old were the rats?
Yeah, good question. So we started with 3-month-old rats, and we took the rats in the NICI group and the NICI control out to 20 weeks. And we took the rats in the MCLT plus MMT group and skin incision control out to 16 weeks.
Okay, so I was having a question. I have a question about, so in your, I think it was the double combination, the MCLT MMT group.
Yes.
Is that correct?
Yeah, that's correct.
You saw a decrease in trabecular number and an increase in trabecular thickness, and you interpreted this as bone loss. Since the surgical procedure occurred at 3 months where the animal was not skeletally mature, could you also interpret this as being a difference in development?
Yeah, you're absolutely right. So we did start with animals that were 3 months old. And in our future studies, we've gone on to start with animals that are 4 months old to reach that level of skeletal maturity, because in the earlier stages, the bone remodeling or the bone modeling kind of outweighs the effects of bone remodeling. So you're absolutely right in terms of that interpretation. Yes, we can. I will say that in this study, because we took the animals out so much further than other groups have in terms of analyzing end-stage OA, we wanted to make sure that the effects that we were seeing were actually due to the increase in severity of the OA and not specifically the age-related changes. And so because we took them out so far and the animals were so old, that's why we came to that conclusion, but that is a good possibility.
Okay, thank you. Nice job.
You're welcome.
Markia, one of the question that was submitted via chat was, what was the load force used to generate ACL? And did you check if there was any bone fracture when you applied the load?
Yeah, so the specific load force, I can get that information back to the person that asked that question, but this was based on the validated study that was done by our collaborators. And one of the ways they checked to make sure that the ACL was ruptured was measuring tibial or anterior posterior tibial draw. And the fracture part of the question, that's always a big risk when we have the non-invasive knee injury, because this isn't a specific cut like we would do in like a surgical procedure where the effects of that cut are so specific and so concentrated on that area. Because we are applying a load to the entire knee, there is always the risk of fracture. And one of the ways that we've verified to see if this occurred has been through the micro-CT. The micro-CT analysis to really look at the microarchitecture of the bone to see if there are any effects of that load being placed on the knee to it. But that's always a risk. So good question.
Yeah, that's always the problem with this model.
Right.
Another question for you, Markia. Did you conduct histopath assessment to confirm end-stage OA?
Yes, so there was histology was conducted on all of the groups. We haven't analyzed that data as yet, but our future studies are looking more closely at the histomorphometric measures to confirm the presence of OA, which is very exciting for our group because we're moving more into the realm of bone work. But yeah, we did do histology to confirm the presence of OA, but that work was not presented here.
Dr. Safadi, you had a question you'd like to ask?
I do. I can't keep quiet, unfortunately. So first of all, it's a great work. Congratulations on your award. I'm always get very excited when I see young generation successful and getting out their work presented. So you just mentioned that you've done some histopath imaging and histology. I don't know if you remember or what you're thinking, the contribution of that bone loss. Do you think basically by invading a pre or well-differentiated osteoclast or decrease bone formation or a combination of both? What are you thinking here?
That's a really interesting question. I think to answer that, the specific type of histopathology we would have to do would probably involve some sort of trap staining that actually looks at the behavior of the osteoclast and the osteoblast in that way. Really here we focused generally on the standard bone histology staining. So we couldn't see the behavior of the osteoclast and the osteoblast in that level of detail. But I think to look at that more closely, we would have to do some sort of trap staining or something to look closely at the remodeling events and not specifically what's happening at that singular time point. So that's something we'll consider for our future work for sure.
That's great. One thing, basically, we've noticed that even the pool of mesenchymal stem cells with a subchondral bone, it's different than if they are within the bone marrow. So if you take MSCs or osteoblasts, osteoclast-derived cells from subchondral bone, put them in culture following the procedure, of course, as well as you take from a bone marrow, they differentiate differently. So you basically, I know you cannot do that now. It's already done with those studies, right. But for future will be interesting to relate what you've done mechanistically as well at the cellular level. So Great work.
Oh, that's interesting. Thank you for that.
Sure.
Can I just follow up on that? So I don't know, you probably are familiar with looking at dynamic properties. So you mentioned looking at trap staining, and you would use Goldner for osteoblasts, but you're familiar also with dynamic properties, will give you mineral apposition rate and bone formation rate across time. That might be something to look.
Yeah, so we're really excited to move into that realm of like histomorphometry. So that would involve like the injections of declomycin and calcine prior to euthanasia to kind of figure out what exactly is happening with the remodeling. So we're kind of excited to start going down that realm. We're currently doing that work with some of our collaborators. So
Okay, excellent.
Great. I'll take one last invite for any other questions.
Just have one comment, one question. How far are you in your training? I hope we're not dumping too many experiments, which make you stay for a long period of time. Probably your mentor might like it, but you might not like it. So I'm just wondering, how far are you with your training here?
yeah, I'm sure you'd like that. I'm in my 4th year right now. And actually, so I'm moving further into the realm of doing more with the bone work outside of the micro CT. So historically, our lab has looked at, we approach OE from a bunch of different perspectives, and I'm kind of the bone person in the lab. So I'm kind of entering that world, and I'm very excited about it. But yeah, I'm entering my 4th year.
Best of luck. Great work.
Thank you.
Great. Thank you so much, Markia, for your presentation. I'd now like to invite our other 2021 URM Mentor Awardee, Ernesto Soloranzo, who is a PhD candidate at Northeast Ohio Medical University, and he will be presenting on the detrimental effects of low aerobic capacity on bone metabolism. So Ernesto, whenever you are ready, you can go ahead and share your screen and unmute yourself.
All right, can you hear me okay?
Yes, we can hear you.
Okay. All right, so I'll go ahead and start. My name is Ernesto Solorsano. I work at Northeast Ohio Medical University. I'm a 4th year PhD candidate. Today I'll be presenting about the detrimental effects of lower aerobic capacity on bone metabolism. Wanted to quickly thank the ASBMR Diversity Committee for inviting me here. And I look forward to your comments. All right, so what is aerobic capacity and how is it relating to bone? So aerobic capacity is a common measurement that reflects on an individual's ability to distribute and use oxygen during exercise. And this has been linked to diseases such as osteoporosis, as well as bone health in general. Aerobic capacity can be adjusted on each individual depending on their exercise regimen. So it's basically a movement measurement and specific to each individual. A high aerobic capacity has been linked to better health outcomes overall in various fields, and while lower aerobic capacity has been a predictor of premature death. But current research on aerobic capacity and its effect on bone is kind of limited, since most of the studies basically look into an animal and make them do an exercise regimen and assess the results afterwards. So therefore, there is a need for a consistent mouse model that will reflect both high and low aerobic capacities. So for this study, we explored the use of these two animal models for high and lower aerobic capacity. We have the lower, they are called the low capacity runners and high capacity runners. Low capacity runners are rat models that, regardless of how much exercise you give them, they're gonna remain obese and really not be able to lose weight and not have very high velocity in the treadmill. In comparison, the HCRs will remain lean regardless of how little exercise they get. So therefore, they are forever lean. So this is a model that will reflect on the inherent capabilities of each individual in regards to their aerobic capacity, and it's known that about 50% of our aerobic capacity can be attributed to our genotype. And consistent with these models, the aerobic capacity matches their high or low running capacity. These rat models have been used in a wide array of studies such as pain, aging, metabolomics, muscle, as well as vascular changes caused by exercise. But sadly, there is still not a lot of work done on bone. There's been some biomechanics work, but really no dissection into how aerobic capacity triggers bone loss or bone gain in these animals. So for this study, we wanted to explore the effect of aerobic capacity on bone health. So for this study, with the collaboration of the University of Toledo with Dr. Lauren Koch, who is basically the only researcher housing these animals and gives them their regular exercise regimen so that they remain with the phenotype in a standardized manner. From here, we acquired two different groups, geriatric group, which are 28-month-old females, and an adult group, which there are 10-month-old females as well in comparison, 6 of each group, for giving us a grand total of 24 animals. All of these animals had the same exercise regimen, at least once a week, where they were put on a treadmill until exhaustion. Obviously, the HCRs would last a lot longer than the LCRs. And for our particular study, once we got tissue from them, we isolated the bone marrow cells, where we isolated mesenchymal stem cells as well as hematopoietic stem cells, while also running micro-CT analysis and bone marker assessment from total bone tissue. So for our hypothesis, we expect the HCR rats to have a higher bone mass than the LCR rats, just to match what's currently known in regards to aerobic capacity. And we also expect the geriatric rats in both the HCR and LCR to have a lower bone mass in comparison to adults. So on our fifth study, we did a micro-CT analysis of both the LCR and HCR, where we can, just by eye, we can see there's a clear difference between bone mass in the HCR group. This can also be reflected on the total bone trabecular volume, where we see HC being consistently higher, and also this is consistent within connectivity. Additionally, we're also presenting the osterics and RUNX2 expression, which are both big osteoblast markers, and these are both upregulated in total bone. So in order to further look into the story of the osteoblast, we collected the mesenchymal stem cells from these animals and differentiated them until completion till week 3. On week 1, we assessed for ALP. Week 2, we assessed for ALP as well. While week 3, we assessed for allosterine differentiation. And just looking at the nodule count, there wasn't a great difference between the geriatric animals, but we do see a stagnant difference between the young and between the young animals in regards to the HCR. And just by in a qualitative manner, we can also see that the adults just consistently had a higher ALP staining while also having a higher mineral deposition. So in conclusion, we state that the osteoblast differentiation was increased in both the adults compared to geriatric, and more in HCR compared to LCR animals. So after this, we decided to look into the other side of the story, looking into the osteoclast, where we isolated hematopoietic stem cells from these animals and assessed them for viability and proliferation. We see a clear difference on the geriatric animals after 24 and 72-hour assessments. We do see a similar trend, while not significantly, different. On the adults, we do not see such a difference. Therefore, claiming that the HCR pre-osteoclast show decreased viability compared to LCR, although it's only consistent on the geriatrics. Later, we differentiated these hematopoietic stem cells into the mature osteoclast, where we did trap staining, and just by eye we can easily observe that, at least from the geriatric ones, we see very little osteoclast differentiation, but this could just be attributed to their geriatric phenotype. Additionally, the adults were able to differentiate them better, and while there wasn't much of a difference between the LCR and HCR groups, this basically is a claim that the osteoclast differentiated there did not show a major difference between the LCR groups, while there was a difference between the geriatric and adult groups. So in conclusion, we claim that the HCR and LCR animals can closely resemble the expected outcome of bone that we would expect from variations in aerobic capacity. Therefore, claiming that this is a good model to further study changes in bone caused by aerobic capacity. The changes in bone volume can be attributed to overactive osteoblasts in these animals, while also a decreased viability at later life. For future studies, we wanted to assess the gene expression of these differentiated osteoclasts and osteoblasts, as well as total bone tissue. We also are preparing to conduct histological and histomorphometric analysis. We also plan to look into the joint phenotype, as well as sending out, as well as checking on the biomarkers for bone formation and resorption in vivo from our plasma samples. In acknowledgements, well, first of all, I wanted to acknowledge all the authors and this project. We spent a lot of time working in the summer, and we had a lot of people involved, so it was a lot of fun. Primarily Dr. Safadi, our collaborator, Dr. Kurch, and most importantly, the ASBMR Diversity and Inclusion Committee for inviting me to this talk today. Are there any questions?
A very nice presentation, Ernesto. So, you know, in my lab, we look at vascular changes in bone. Have you guys considered that, or have you looked at anything?
Actually, vascular changes have already been studied on these animals before. I don't know if they did specifically in geriatric in comparison to adults, but they did see, because they looked specifically into the oxygen intake, they had more vascularized muscle tissue on these animals.
In muscle tissue, what about bone?
In bone, no. There is no studies conducted in that regard.
Have you guys considered that possibility?
Oh, definitely. Again, we will be doing a lot of work with the histology analysis, at least.
Okay.
This is Fayez here. Maybe we'll send him to your lab if you're
Yes,
interested.
definitely.
There you go. Expect the shipments then.
I'd welcome it.
That's great. That's great. Thank you.
I have a question. Yeah, so Ernesto, yeah, this was a wonderful talk. Thank you. It's a really interesting study. So yeah, I have a question about, so you had mentioned in your talk that I think you had mentioned, 50% of the sort of aerobic capacity in these rats can be accounted for by genotype. And so I was wondering if, what's known as far as specific genetic variants that are different between the LCR and HCR rats, and whether that can inform sort of biological mechanisms by which you're seeing these different responses in the bone.
Oh, that's a great question. So the 50% data that I mentioned this is based off of human work. So it's basically most of the work has already been done on human, but in order to be able to replicate it, they're trying to pretty much use this model for that. Going back to your question in regards to genetic analysis, not a lot of work has actually been done on that, because these rats were just specifically bred, selectively bred when they were, these ones retain more fat on the treadmill in comparison to the other ones. So yeah, in regards to the genetic analysis, there's a lot to be done. We'll be excited to look into that.
Well, thank you.
Mm-hmm.
So Ernesto, do you think, since 50% is contributed to genetic, I guess, genetic components, Do you think that can you train the low capacity runners to then have VO2 maxes similar to the high capacity, and then maybe you'd see changes in bone volume?
Yeah.
Is
So this has actually also been done on these animals. So they put them on a treadmill, and the low capacity runners will have an increase similar to the high capacity runners as well, but they will quickly revert back to what they started at. They will not retain this aerobic capacity for very long if you keep them, let's say at a lower exercise regimen, while the high aerobic capacity ones are more likely to keep this for longer.
Do you know how many weeks it would take to get the low capacity runners up to the VO2 max levels as the high capacity? Do you remember from the experiments in the
I don't remember exactly how many weeks, but I can let you know later. That's okay.
I just wanted to, Marquia and Ernesto, very nice presentations, both of you. And I also just wanted to remind you guys to add this seminar invite to your CVs.
Will do.
I didn't actually think about that. Let me see.
Okay, if there are no other questions, but please feel free to submit a question in the chat if there is any other additional ones. But at this time, I'd like to take the opportunity to thank both Ernesto and Marquia today for taking the time to present your research and really great talks. We really appreciate your participation in this special spotlight session. And a recording of this session will be posted on our ASBMR YouTube channel and the link available on the ASBMR website. Our next spotlight will actually be next Wednesday. And I just want to pull this up on November 17th at the same time, 1 p.m. Eastern. And we will be hearing from Alicia Diaz-Thomas and Rodrigo Valderrama Valderrabano on their different research projects that they have been working on recently. So I do please invite you to join us again next Wednesday at 1. And thank you everyone for your time today. We really appreciate it.
Bye, everybody.
Thank you.